So, welcome everyone. It's a pleasure for me today to open this second European Mineralogical Union public lecture of this year. As you probably know, the Council of the European Mineralogical Union usually asks the recipients of the Research Excellence Medal to give public lectures. And on 2025, the recipients of the medal are Alberto Vitale Provarone and Julie Cosmidis. Professor Provarone has already given his public lecture a couple of weeks ago. And today, it's a pleasure for me to introduce you the second public lecture. And in particular, I would like to introduce, first of all, the recipients of the medal and the speaker, Julie Cosmidis. A few words about the vita of Julie. Julie Cosmidis has obtained her PhD in 2013 in Paris, in France. And after 2 years of postdoctoral studies at University of Boulder in Colorado, she joined the Pennsylvania State University as an assistant professor. And later on, in 2020, she returned to Europe as an associate professor at the Oxford University. And using the words of the evaluating commission for the Research Excellence Medal, Julie Cosmidis has emerged as a leading figure in the field of biomineralization at international level by combining leading-edge experiments in mineralogy and biochemistry and nanoscale investigation to constrain the driving forces of biomineralization. For the major impact of her numerous achievements, Julie Cosmidis is truly worthy to receive the EMU Medal of Research Excellence 2025. And so, Julie, the stage is yours. The title of the presentation that Julie is going to give is Decoding Carbonate Biomineralization using high-throughput mineralogy. Please, Julie.
Thank you very much, Diego, for the kind introduction. And thank you everyone at the EMU for this medal. I'm really honored. Can you see my slides correctly? Yes? Okay, thank you. So before I start, I will spend a minute thanking all the people really who have helped me different stages of my career. So I'm thinking about mentors and colleagues and collaborators and friends, but especially the students and postdocs, because as you know, as a mid-career scientist, spend most of your time responding to emails, and these people are in the lab doing amazing things. So this medal really should go to them as much as myself. So 2 weeks ago, we had a really nice presentation by Alberto, and we had a lot of beautiful pictures of mountains and outcrops and so on. And although I'm not very much a field scientist, I thought I should show at least one geological image. So this is what you see if you take a ferry from France, where I grew up, and you go towards England. And you will see those beautiful cliffs, as you know, they are the White Cliffs of Dover. I tried to find a picture that was representative, especially of the weather you can expect to find as you approach Britain. And those cliffs, of course, are made of the fossilized skeletons of microscopic algae. So the fragments of skeletons are called coccoliths, and they are composed of calcium carbonate, which will be the topic of this talk. And they are biominerals, so they are minerals formed by microbial or biological activity. And they are probably the most abundant type of biomineral on Earth. And they're interested for geochemists because if you consider their accumulation over a geological timescale, and if you combine their formation with continental weathering, you have a very important carbon sink, which is an important process for the geological cycle of this element. And Biominerals can be formed by eukaryotes, like algae and animals, but they can also be formed by prokaryotes, so what I will call microbes in this presentation, so bacteria and archaea. And they are most likely also the most ancient type of biomineral on Earth. If you look at a typical Archean landscape, we have here a beautiful artist view of what an Archean landscape may have looked like. You can see in the foreground, you have these round sedimentary structures that we consider are among the oldest and the most robust traces of life on our planet. So for example, here we have the almost 3.5 billion year old stromatolites from the Dresser Formation. And we know these objects, these structures are formed by bacterial biomineralization because we have modern equivalents today. For example, the very famous Shark Bay stromatolites. And these objects have been studied for a long time now, and we have some idea about what is the mechanism for this microbial biomineralization process. So microbes, of course, they're alive, they have physiology, they exchange elements with their environment. And as a result of these metabolic reactions, they modify the chemical environment around the cells. And we know that some microbial metabolism, so both oxygenic and anoxygenic photosynthesis, along with sulfate reduction, can drive the chemical environment towards supersaturation with respect to the calcium carbonate. So there's different ways you can represent that, but basically they locally increase the pH and alkalinity and can cause calcium carbonate to form. And we know these specific metabolisms are indeed abundant in stromatolites because people can go in the field, collect microbiolites, extract DNA, sequence them, and look at the diversity of microbes in this type of environment and identify these different metabolisms. So this is an example, a really nice study by Miguel Iniesto and collaborators. There's another more passive process that we think is involved in the formation of those minerals, which is the fact that the microbes, they have, of course, cell surfaces. They also produce in the extracellular environment a lot of organic polymers called EPS that can act as surfaces for the nucleation of the calcium carbonate. So if you take a solution that's supersaturated with respect to those minerals, it's possible that you won't precipitate any because there's a kinetic barrier that needs to be overcome to allow this mineralization process to start. But if you have a pre-existing surface that can act as a template for the nucleation process, you lower the kinetic barrier and you allow what we call the heterogeneous nucleation to occur. So we think if we look again at microbiolite, we can see we have kind of textural evidence that the mineralization process starts at the surface of the cells and on their EPS. And so that's a more passive process through which biomineralization can occur. So there's a bit of terminology on this slide. If you're not a specialist in this field, you don't need to worry about remembering those terms, but we call the first active process I mentioned induced mineralization, and the second passive one influence mineralization. There's a third type of carbonate mineralization process I won't be talking about in this talk that we call microbially controlled mineralization, where the cells actively pump calcium and other cation within the cell, and they grow amorphous calcium carbonates often within dedicated vesicles inside the cells. And I'm mentioning them mostly to show this beautiful image here of a cyanobacterium packed with this amorphous calcium carbonate. And I encourage everyone, if you are interested in this process, to go, for example, read about the work of Karim Benzerara, who has been studying this process for many years. So the question I want to focus on in this talk is what controls the properties of biominerals. So when you look at any mineral, it has a set of properties such as the size and the shape of the crystals, the texture, the localization, is it intracellular or extracellular, the crystal structure, the chemical composition, so elemental composition, isotopic, also presence of organic molecule. So I call all these properties the mineral phenotype. And the question here is really trying to understand what processes control all these important properties. And this is an important question when we want to use minerals as biosignatures, for example. So bacteria are very small. They are also made of organic molecules. They are soft bodies, so they don't fossilize very well. However, when they form minerals, they potentially can be preserved in the geological record and used as evidence for life, for example, on early Earth, but also potentially on other planets, right? So here I have pasted like 2 snapshots of paper that specifically discuss whether we can use biominerals as potential biosignatures, right? And if we want to be able to do that, of course, we have to determine whether the properties of the minerals are distinct from those of abiogenic minerals that would form in specific environments, right? And if we find special properties in the biominerals, then we need to understand what they are a signature of. For example, here, this is a scanning electron micrograph of calcium carbonate minerals. They have indeed been formed by microbes in an experiment. And you see that very weird morphologies and textures and that are potentially can be used as biosignatures, but we don't know a signature of what, right? What is causing these special properties? Is it a result of chemical changes induced by the microbes in their environment? It is a result of specific organic molecules that they are produced? Is it really encoded into genes? This kind of question. We need to know what these properties are a signature. Another reason why we are interested in understanding what controls this mineral phenotype and also being able to control it ourselves is for applications of carbonate biomineralization. So there are different proposed practical applications for microbial carbonate minerals. One of them is in construction. So some people now use bacteria to make biocements and bioconcrete. There's a student in my lab at the moment, Alan Source, who is a visiting PhD student who is making self-healing concretes. So he's making concrete samples that have encapsulated bacteria. And whenever the concrete cracks, the bacteria which were dormant can spring back to life and start biomineralizing and filling the crack with carbonate cement. There's also some applications for what we call ex-situ carbon mineralization, where if you react some alkalinity that's coming from some solid waste with CO2, you can use bacteria to convert the CO2 into calcium carbonate particles that may have different practical applications. So you can, downstream of this process, sell your carbonate particles to different industries. And what you see that these different applications, they require that we are able to control very well the biomineralization process and also to control the specific mineral properties, because those properties are going to determine whether or not the particles can be used for different downstream applications. So I won't talk too much about those applied topics. I just will mention that we had a project funded by Innovate UK a few months ago, where we worked with a company called Cyanocapture, trying to screen different strains of cyanobacteria for this specific carbon capture and particle manufacturing processes. So I will start by a case study where we're going to try to understand what controls biomineralization process by looking at an example in the field. I've been working on a site in New York State in the US that's called Green Lake. And every spring and summer in this lake, you have what is called a whiting event, which is the massive precipitation in the water column of fine-grained calcite particles. And this site has been studied since the 90s, and it has been established that the whiting coincides every year with the bloom of a cyanobacterium called Synechococcus. So whenever cyanobacteria are blooming, you have this whiting event. And that fits well with the model where the cyanobacteria, through their photosynthetic activity, they trigger supersaturation with respect to calcium carbonate and precipitate those minerals. You can also isolate Synechococcus from this environment and inoculate it in a sterilized sample of the lake water. And if you do that, you can see that the bacteria, they trigger the formation of the minerals, and specifically the calcite starts nucleating at the surface of the cells, almost specifically on a layer of proteins called the S layer. So this is this heterogeneous nucleation ID. Again, you can demonstrate it with samples from the field. A few years ago, Chloe Stanton was a student I was working with at Penn State, revisited Green Lake and collected samples of the whitings. And she could demonstrate that the whitings coincides with this bloom of cyanobacteria, as has been established before, but also with a bloom of diatoms, which are also very abundant at the time of the whiting. And what we could see on these images as well is that the calcite particles, they are present within a mesh of organic polymers. You see there are these threads you see on those images that are EPS, so extracellular polymers, most precisely chitin, that are produced by the diatoms. So we were wondering if maybe diatoms had not been overlooked in previous investigations of Green Lake. Chloe had samples from early in the whiting, and she took them to the synchrotron to use a method that's called scanning transmission X-ray microscopy, or STXM, that allows us to map at a very fine scale the distribution of organic carbon in red here. You can see bacteria, for example, and calcium in green or blue. And what Chloe could find is that calcium was mainly present at the surface of the diatoms and their EPS, which seem to be very good at absorbing this calcium. And what we could also see is that within the EPS around the diatom, you could find those first particles, those first nuclei of amorphous calcium carbonates, again forming as a first stage early in the whiting. So this was a nice new model. Okay, maybe the diatom, through their photosynthetic activity and through the production of this EPS, could play a role in the formation of the calcium carbonates. So last year, with a master student at Oxford, Mbere Umo, we wanted to test this model. So we purchased 2 commercial strains of diatoms, Cyclotella cryptica and Thalassiosira rotula, which are known to produce large amount of chitins. So these are those fibers you see that are being produced through these spores, through these pores of the skeletons. And the experiment is to inoculate those diatoms in a medium that was supersaturated with respect to calcium carbonate, as is the case of the water in Green Lake. So we had actually 2 sets of experiments, one that had low supersaturation level and one that had a much higher supersaturation level. And what we could see in those experiments is that the diatoms, they initially increase the solution, the pH of the solution, most likely due to their photosynthetic activity. So they were pushing up supersaturation with respect to calcium carbonate. But if you look at the calcium curves on the right, so they show the evolution of calcium in solution with time, you can see that you don't really see a difference in the intensity of the mineralization in the abiotic controls in black or with the diatoms in red and blue. So diatoms modify the environment, but eventually that's not a very important parameter controlling the amount of mineralization, at least, because you see the same amount of precipitation in the abiotic controls. So chemically, there's nothing really very interesting going on. But now if we look at samples that are collected on filters and we look at them with scanning electron microscopy, we can look at the morphology of the particles that are formed in different conditions. So on the left, you have an abiotic control without the diatoms. And in this case, the minerals are prismatic calcite particles. So we determine the mineralogy using Raman spectroscopy. And on the right, you have the particles formed by the diatoms. And we can see that in one case, with T. rotula, we're forming calcite cauliflowers. That's how we call them. They're these complex particles. And we also have some spherical carbonates, which are the vaterite polymorph. And on the right, with C. cryptica, we also have the calcite cauliflowers, but we also have aragonite needles. So we can see that the diatoms, although they don't seem to influence much the total amount of carbonate we form, they do seem to play a role in controlling the mineral phenotype, so controlling the crystal structure and the morphology of the particles. And when we see that, there are many questions we can ask ourselves. So are those differences in mineralogy due to difference maybe in the bulk solution chemistry, and we don't see much difference, maybe in the local solution chemistry around the cells? Is this due to heterogeneous nucleation on cells or the EPS? Is it due to other type of organic mineral interactions? At this stage, we don't really know from just looking at these images. So there is an approach we like to use in my lab that we call in vivo microscopy. So rather than growing cells in a tube and then extracting sample and preparing them for, let's say, SEM, for example, we try to do observations directly in vivo in the live culture. So we grow the cells in transparent multi-well plates. For example, here it's a 96-well plate. And we use inverted microscopy to image the bottom of the wells and see the minerals form as the diatoms are growing. So in this image, for example, the larger, darker particles we see are calcium carbonates, and the smaller ones that tend to form little chains, they are the diatoms. And we can see, the minerals forming in real time using, for example, time-lapse imaging. So if I zoom in an area of a well, this is what it looks like. So on the left, you have a bright field image. You see the calcium carbonate particles. They are the large minerals, and the small ones are the diatoms. And we can also use, on the same field of view, fluorescence microscopy. And on these images, only the diatoms light up because their pigments make them autofluorescent. And from taking a quick look at an image like that, it seems that the minerals are forming preferentially on or around the diatoms, right? If you look at some of them, you even see diatoms seem to be included into the mineral. But on the other hand, well, there's a lot of diatoms, so we don't know if the carbonates were forming in random locations. Maybe they would appear as if they were forming in association with the diatoms, right? So it's hard to see just from an image. But you can do some processing on these images. So here what I did is that I measured the mean distance between the edge of a carbonate and the edge of the diatoms. So this is the red line on this graph. And then I ran a Monte Carlo simulations on which I simulated, okay, if the carbonates were randomly distributed, can we compute the random distance between the diatoms and the minerals? And you can do, I did thousands of such simulations. So this is the graph bar on the right. These are the results of the Monte Carlo simulation. And you can see a very nice discrimination. The p-value is like 0. It's really statistically significant that the carbonates are much closer to the diatom than they would be if it was a random process. So it is a very complicated way to explain that here we have evidence in kind of a robust way, because it's all observed in situ without having to disrupt the experiments, that organic mineral interactions are likely important in this process. So this case study here hopefully was or can lead us to have this conceptual image here in our minds of all the different processes that may influence the mineral phenotype. So if you look at a cell making a minerals, be it intracellular or extracellular, there's a number of parameters that can control what the mineral is going to look like and what its properties are going to be. So there's going to be the chemical environment, of course, so the composition of the medium, the temperature, things like that. And then there's probably going to be some interactions between biomolecules, for example, the cell surface or the EPS or soluble organics that are in the extracellular medium that may also have an effect on the mineral phenotypes. And of course, those biomolecules are encoded into genes that are part of the genome of the microbes. And those genes are also encoding the physiology of the cells. So in which ways it's transforming the chemistry of the medium. So we have a very complex picture here. And I'm going to go back several times in this talk to, and trying to zoom in at some of these aspects of this picture and see how we can address this complexity. The one thing I wanted to stress on here is that organic mineral interactions are very important if we want to understand biomineralization process. And it is also true in the case of uncontrolled biomineralization. Okay, so sometimes people have in mind that it's only in controlled biomineralization that is important to understand the role of organic biomolecules. I want to argue that it's also the case of this other uncontrolled biomineralization process I talked about in the introduction. And you can show in very simple inorganic or abiotic experiments, the huge effect that organics can have on the properties of carbonate minerals. So if you precipitate a calcium carbonate, for example, calcite, in an inorganic, very clean medium, and you let time for the crystal to grow, you may obtain this perfect rhombohedral calcite, which morphology reflect the unit cell. So it's very simple. And here I want to show you some images from the literature where people have precipitated carbonates in the presence of different organic molecules. Some of them very simple, like citrate, or some of them very complex. And you can see that just the presence of these organics can give rise to these very complex crystal morphologies, right? So there's a very strong influence of organics in solution on the morphology and the structure of the carbonates. My favorite one is the one at the bottom right, where if you use a chiral molecule like D-aspartate, you may obtain a chiral carbonate crystal, which I find very cool. So there's many different ways in which organics can exert a control on the mineralization process and alter its structure and its morphology. So here I put them all in one slide. I won't go through them. There is a really good reviews by Meldrum and Colfen that I invite you to read if you're interested in those things. I will just say that these mechanisms are multiple and they are very complex and very hard to model and predict, which means that if I design a mineralization experiment and I introduce an unknown organic molecule, I have no way to predict what will be the shape and the structure of the resulting calcium carbonate forming in the presence of this molecule. So I'm going to explain now how we are trying to better understand these interactions. So in the experiments I'm going to describe here, we're focusing on abiotic systems, so without any microbes, but we're working in systems where we vary some environmental parameters, so the composition of the medium and the presence of different soluble organics in the medium. So in a system like that, very simple, you have an aqueous solution, and there's a range of parameters that you can vary. Of course, the temperature, the pH, the total amount of inorganic carbons, the DIC, can vary alkalinity. This is going to result in variation in the supersaturation level. You can introduce some ions like magnesium, phosphate, and sulfate, which are known to influence mineralization, trace elements. So there are a lot of chemical parameters that could be important in this process. And of course, we're adding pure organic molecules that we call organic additive. And we do this mineralization experiment And after minerals are formed, we can look at them and characterize them and characterize different properties of the particles, so their size, their morphology. We could also look at their composition. In case they contain magnesium, it's possible to measure the ratio of magnesium over calcium, for example, and the crystal structure, so in particular the polymorphism. Do we have calcite, aragonite, vaterite, amorphous calcium carbonate, or something else? And the big question here is, can we predict the properties of the carbonates that are going to form under a specific set of environmental conditions, including different organics? And the short answer is, it's impossible to do in the current state of things, right? So a classical study on this kind of question would be taking a dozen or a few dozen of tubes and varying some parameters. So typically in this study, it's 1 or 2 parameters, letting minerals form, and then extracting or sampling these minerals and looking at them with different methods. So that would typically be XRD to look at the crystal structure, maybe a bit of microscopy to determine the morphology. And of course, the studies are a bit slow to make, so that's why typically you would only look at 1 or 2 parameters at a time and obtain often a plot like this. For example, at the bottom left, you have the magnesium over calcium ratio in the solution on one axis, the temperature at the other one, and each dot is a distinct experiment. And the authors have determined, is it calcite or aragonite? And they try to find the limits between those two faces in this space to find in which condition you form one or the other. The problem is that it's very hard to extrapolate results from one study to the other. Okay, so if you take the first figure that looks at temperature and magnesium, and then you look at the second one that looks at the effect of sulfate, for example, the first study didn't have any sulfate, and the second one maybe was working at a different temperature that was not included in the first study, or wasn't looking at magnesium, or didn't change the calcium concentration. What I mean is that it's hard to incorporate those results from this study in a single model. And then you add another one, and you have a series of results really that are hard to extrapolate from one study to another. And this is how I see this complex problem, right? So let's say you're only looking at 2 parameters, so you have chemical parameter 1 and 2, and you vary them, and each of the dots in this image is a different experiment. And you determine that, for example, you have the red dots are calcite and the blue ones are vaterite, for example. It's quite easy to find an equation for the line that is going to separate those 2 domains in a 2D space like that. And maybe you can add a third parameter, right? So you vary another component of the solution, and now you have a 3D space, and now you have to find the equation of a plane in this space. It's still doable at the empirical level, and then processing the data and finding the equation for this plane. But what if now you add a 4th parameter, or a 5th, or a 6th? You have a multi-dimensional space, and now it's impossible almost to, at least visually, to find an equation like that. What if we add an organic molecule, right? We've seen the dramatic effect that organic molecules can have on the morphology and structure of the particles. What if we change the concentration of the organic molecules? We see that we end up with a very complex system in which it's very hard to make any prediction. So in a space like this with a very high level of multidimensionality, it's very hard to make any prediction at all and relate some input parameter to an output in terms of mineralogy. Machine learning is very well equipped to deal with this kind of problem. So if we look at a predictive or classification machine learning algorithm, really what it does is that it learns to recognize outputs for a set of input parameters, right? And it's going to work provided that you have enough data to train this algorithm and teach him, okay, this is the output we have when the set of input parameter looks like this. The problem is that if we want to build this kind of machine learning models, we of course need a lot of data to train the model. So I'm getting to the main method which we've been working on as a part of an ERC starting ground called BioFacts. So my postdoc Luca have been working on methods that allows us to significantly accelerate the rate at which we can do mineralization experiments and characterize the output or the mineralogy of the minerals under different conditions. So he uses this 96 well plate setup. So each well is a different mineralization experiment. And you can vary different geochemical parameters. You can vary different organic molecules. And after mineralization has occurred, we can then characterize the minerals that are formed in those different conditions. And Luca has a workflow that has 3 steps. I'm going to go through them, but the first step is looking imaging, so microscopy, to get information on the particle morphology. In the second step, we classify the particles into different types. I'm going to explain why we need to do that. And in the third step, we go to different classes of particles and do Raman spectroscopy on them, which allows us to get mineralogical information like calcium carbonate polymorphisms, for example. So I'm going back to the first step. So we're using an automated high-resolution inverted microscope. So you have your plate, and the microscope is able to automatically go on different wells of the plate and focus on the bottom of the well, which is where the particles are, and acquire images for the whole well. So you can image all the particles in all the wells in your plate. And then there is image processing, automated image processing to detect on the images the different particle and outline them, so you have a contour, and then extract a range of what we call morphometric parameters. So they are numerical parameters that are quantifying the morphology of the particles, so their size, their area, perimeter, and so on, but also quantifying what their shape is. So we have parameters like aspect ratio, eccentricity, circularity, and so on. I think like 75 different parameters we can calculate automatically so that the morphology of each particle is quantitatively characterized. So with a system like that, what you can do is very rapidly identify morphological changes in your data set as you change some chemical parameters. So these graphs here are just distributions of different morphometric parameters for 3 different experiments. And you can very rapidly visualize how the composition of the medium is influencing changes in the shape of the particles, for example. The second step of the pipeline is particle classification. So very often, or typically, in a data set, let's say in a plate, we see that we find only a limited number of different particle types. So on the images here, for example, on the top left, you have 2 main types of particle, right? You have those that are in red here, type 1, which are a bit irregular, and you have the type 2 that are more prismatic. So Luca identified, based on his first 300 carbonate mineralization experiments using 23 different organic additives, that he could identify 10 different types of particle in his experiments, and each of them systematically correspond to a different crystal structure. Right? So using that, he's been able to design a machine learning model called random forest model that now allows him to, based on the morphometric information extracted from step 1 of the process, automatically classify the particles that are present. So an output of the program would be, okay, in well 1, you have 20% of type 1 particle and 80% of type 5 particles, for example. And these are just figures that show you that classification is working well with a 96.7 accuracy. And the figure on the right is called a feature important analysis. It tells you which morphometric parameters are most important for the classification. So an example of application of this method here, for example, these are just 6 experiments where we varied the calcium concentration. So you have 2 different calcium concentrations and the concentration of an organic molecule, an amino acid called asparagine. And on the top chart are showing you the total number of particles forming in these different experiments, and then the total particle surface area, so the total area covered by particle, which is a proxy for how much minerals are formed. And it's showing us, for example, that the influence of the amino acid on the particle count and total abundance depends on the calcium concentration, right? So at low calcium concentration, the more you have amino acid, the more particles you form. But when you are at high concentration, it's the opposite effect, right? So, but what is interesting here on the bottom part is showing you different particle types in these different conditions. So what we see that at low amino acid concentration, regardless of the concentration of calcium, we mainly find type 10 particle and a bit of type 1. Now, if we increase the concentration of the amino acid, changing the concentration of calcium has a huge effect. At low calcium, you have a lot, or exclusively particle 1, and at high calcium, you have particle 6 and 7 that become more abundant. So this is just to give you an example of the kind of relationship you can now start investigating once you have a system that allows you to do many experiments like this. The third step of the data processing pipeline is Raman spectroscopy. So this is just a slide that explains Raman spectroscopy very quickly. When you hit a molecule in a sample with light, this light is scattered by the molecules, and most of the light is elastically scattered, which means it hasn't changed its wavelength of energy. But a small fraction of the light will have changed energy, so it will either gain or lose energy, and this is what we call the Stoke and the anti-Stoke scattering or Raman scattering. And if we look at the specific wavelengths of the scattered line, we have a way to know about the different vibration modes in the material. So know what type of molecules are present, but also what the crystal structure, because different crystal structure of the same mineral will scatter light at different wavelengths. And the reason why we really like Raman for this project is that Raman can be combined with microscopy. So in a confocal microscope, you can do Raman and focus the laser on specific objects that we are observing with the microscope. So it's spatially resolved. It's a very quick measurement, but also the sample preparation is very easy because you don't need a dry sample, you don't need a powder, you can work directly in liquid. So this is what we call in-situ analysis. And the Raman is also not destructive to cells if we keep the laser power low, so we can also do in vivo analysis. And this is just an example of study by my former student, Chrissy Nims, where she was able to look at minerals inside live cells and look at different polymorphs of sulfur minerals inside the cell. So it's really well suited to the setup we have and be able to do in vivo mineralogy. So the only problem with Raman, if it's much slower than the microscopy, right? You can image a whole well in a few seconds, but if you have to analyze one by one all the crystal in a well, it's going to take hours. So if you want to do the whole plate, it will take days. This is where this classification step is helpful, because once you have identified which classes of particles are present in your plate, you only have to go sample a few representative examples of each type and know what is their mineralogy. And we have a very consistent relationship between particle morphology and particle composition, so crystal structure. So we only need to do the Raman on a very small subset of each particle to know about the mineralogy of the whole plate or the whole dataset. So with this method, we can rapidly get very important amount of data. And this is just a figure that shows you how you can visualize a whole data set. On the left, different experiments where you can plot the size of the particle on the y-axis, the number of particles on the x-axis for these 300 experiments, and see, okay, if I want to target small particles, these are the organic additives I want to use, for example. And on the right, you can also plot your individual particles and see that you have a massive amount of data you can acquire rapidly. So the next step with this work, and this is a project that was recently funded by NERC, is to use all this data and acquire much more to build the machine learning model that are going to be able to take as input feature the composition of the medium, the organic molecule at different concentration, and try to predict what will be the mineralogy of the carbonates in these different conditions. So I will be looking for 2 postdocs for this project. So one to start this fall on the experimental aspect, and the other one a year later on building the machine learning models. So if you are interested, please get in touch. So I'm going to finish with going back to biology now. So I was talking about abiotic experiments for a little while, but now I'm going to present experiments where we introduce bacteria in those wells, and we try to understand the effect, again, of the environment, but also of the cell physiology on the mineralization process. So this is something my postdoc Mohamed Ramanji is working on. He's been working with a bacterium called Sporosarcina pastori, which is a ureolytic bacterium. So its metabolism involves the hydrolysis of urea, which strongly increases pH in the environment and triggers calcium carbonate precipitation. And Mohamed has been doing like a huge amount of experiment where he varied a lot of parameters in the medium. So the calcium source, its concentration of urea, concentration of nutrients, cell density, and the temperature. So this is just to give you a snapshot, for different calcium sources, different temperature, different concentration of calcium, representative images of what you observe in these wells. And what Mohamed is trying to do with this data set is to build models using a methodology that's called response surface methodology, where you can visualize as a function of 2 parameters here, for example, concentration of calcium and temperature, how much particles, for example, you can expect to form in those cultures. So, but building these models require you have enough data to calculate those surfaces in this 3D space. Using this classification algorithm, you can also visualize the different types of particles, again corresponding to different polymorphs or calcium carbonates, where they are forming in those different experiments. So line 1 and 3 on these graphs are abiotic control, so experiment without any bacteria. And you can see that we mainly form the type 1 calcite particles. And on line 2 and 4, we have experiments with microbes. And we can see that depending on the conditions, sometimes we form a lot of particle 5 particles. Sometimes we have a mixture of many different types of particles. So again, it's a rapid way to visualize how the composition of the mediums and the cells are influencing the minerals. We're also looking at the effect of cell density, trying to get to this physiological effect that the cells have. And of course, we can see that the more cells we have, the more dense the culture is, the more particle we form, the higher the yield of mineralization we have. And we can see similar effect with the concentration of urea and nutrients. So now the game is trying to disentangle the effect of the chemistry of the medium from the physiological effect of the cells to explain those differences that we find. If I have a bit of time, I'd like to finish with what we're doing to address the genetic of the process. And for this, we're using an approach that's called mutagenesis and screening. So we work with a strain of bacterium, and we obtain what's called a mutant library. So we mutate the genome in many different locations. And then we screen this library to identify mutants which have a different mineral phenotype. So the mineralization process that changes in any way. Maybe it's not mineralizing at all, maybe the minerals look different. And then if we take those mutants and we sequence them and we compare the genome with that of the wild type, we can identify new genes that are influencing the mineralization process. So Mohamed, again, is working on building a mutant library for S. pastori. You already have 71 different mutants. And then using the method I presented today to screen this library and identify mutants that produce minerals that look different. What we find is that we really need to control for the effect of cell density because mutants grow at different rates, and this has an important effect on the mineralogy. So we need to work to disentangle these different effects. So that is very much work in progress. And yeah, these are my conclusions. Sorry if I was a bit long. It's just to say, sorry, how do I go back? Well, maybe I will stop sharing. It was just to say that mineralization is complex, and we're hoping we can use these new methods to better understanding the different parameters that are important. Thank you for listening.
Okay, thanks a lot, Julie, for your very nice presentation. And I would like to ask, we can open the discussion session. I would like to ask if there's any question from the audience. We have also to check if someone writes something in the chat.
Yes.
Actually.
Then I can maybe I can start.
Okay, Isabel, please.
I'm the one in charge of the questions. It was very, very intriguing to see the relation between the variety of the phenotypes of the carbonates that you've seen that really seem to depend on the amount of calcium that you put in the system, even in the abiotic experiment. So one may have thought that, well, when you involve really the cells, when there's a low calcium concentration, then it will stop with one phenotype. And then when it can not accommodate this one anymore, it has to do some various, adopt some different strategies, but looks that it's very similar in the abiotic experiments. So I was actually wondering if you had a chance to look at these aspects in terms of the energy that's required to crystallize the different forms of calcium carbonates.
Yes, so I don't know how I would start. You mean calculating activation energies for different structures and maybe different morphologies
and size and
yeah. What type of method would you use? I have no idea how I would do that. I'm curious to
well,
hear.
start maybe with the surface energy.
Yeah.
That's an endpoint point. That might not be the final answer, of course.
Yeah.
Yeah, an entering point.
Yeah, because if you have a very high surface area versus a particle that has smaller one, you would have a smaller surface energy that tends to be destabilizing. So maybe you can, that can be a way to predict what will form in different conditions. Yeah, I will think about that. Thank you, Isabel.
We do have questions.
Yes, there are a couple of questions from the chat. The first one is, won't the bacterial EPS chelate the calcium?
Yes, so yeah, absolutely. So there's the effect of the EPS is a bit complex because in one way they chelate the calcium, so they lower the amount of free calcium available for the precipitation. So potentially when you have EPS, you're less likely to precipitate. But I think at some level, if the calcium is still above supersaturation, then this adsorbed calcium at the surface is very important in heterogeneous nucleation, right? So you have this dual effect of the EPS that you need to think about. Yes.
Okay. There's a second question for you, Julie, from the chat. And this is, does amount of soluble organics increase the speed of mineralization? If so, what are the common features of these organics?
Yes, so I think so the speed is something I didn't talk about at all, but this is a question we can address with this method of in-situ microscopy, because we can do time-lapse imaging. So we can acquire image on a single well at different time points. And I decided not to include a movie about that, but I should have maybe. And you can see the minerals growing, and you can measure how fast they are growing with time, and start to see how different conditions affect the speed of mineralization. to be honest, I don't have the data set in mind to answer the question, does it speed up mineralization or does it slow it down? Maybe it depends on the organics. But really our goal with this project I presented, where we're going to use machine learning to process this data set, is to find what are the common features of organics that influence the most the mineralization process. So if you tested hundreds of different organic molecules and you systematically determine whether they speed down or accelerate mineralization, then you can identify what is the common features in these organics that are either playing the role of inhibitor or accelerator of the process. So hopefully in a few years, we'll be able to answer this question very precisely.
Okay. There's an additional question from the chat, and then there's one from, I think someone who wants to ask a question directly. The last from the chat is, would you expect that mixed cultures would have further major effect on the characteristics of the precipitating minerals?
What I think we can say is that the more complex the environment, the more complex the minerals tend to be. Either they have more, complicated morphologies, or we form more than one type of minerals if we introduce bacterial surfaces and EPS. So maybe in a very broad way, if you have a diversity of microbes, especially maybe if they have different types of physiologies or they produce different type of organics, you could have a more complicated mixture of minerals. But we're not working with mixtures or communities of bacteria. We're really focusing on single strain at the moment, so we can better understand the system and have it we can model it more easily.
Okay, we can go to the question. So please, you can switch on your
Basultan, you may switch your mic on if you would like.
Thank you so much for great presentation here. So I have question like the slide that you mentioned before about evidence for cell mineral interactions. Some diatomics here not interact with calcium carbonate, or like they separated only some diatomics that show in the electron microscopy interact with calcium carbonate. So my question is, how about other diatomics cells that not interact with calcium carbonate? Does it mean the source of calcium or carbonate lack? Because or is there something missing with the cells that they cannot make calcium carbonate by themselves?
Yeah, that's a good question. So in these images where we see the carbonates, and they seem to be originating from like one diatom, and another diatom won't have any carbonate associated with it. So yeah, the obvious question is why why does nucleation happen there and not another place? I honestly I don't really know. I mean, maybe some are more active than others, like they are, changing the environment more. There's a bit, nucleation is a stochastic process, right? So it's a bit random where it starts. What we can see from other experiments is when you have a mineral that grows, right, it consumes the calcium and the carbonate ions around it. So the local concentrations of these ions, around this crystal are much lower. So it's less likely than another crystal will form there. It will form at a distance where the concentration of its ions are higher. So maybe it's not possible for the diatom really close to a mineral that has already formed to also form a mineral because the local concentrations are much lower. So that's maybe also a factor that can explain those differences.
I see. So what about you said before that you also make experiments with higher concentration of calcium, or if I'm not mistaken, like is that improve that intensity of minerals occur by diatomics?
Yes,
By
yeah, the general rule, the more calcium you have, the more mineralization you will get. That's really an important driver. If you have enough carbonate ions present, that's an important driver of the process. Yes.
Okay, thanks. We can go ahead. Damien Fave.
Yeah, thank you for the talk. I was actually wondering, so the nucleation, as you say, is a dynamic process. So when you decided to present your result, is it always at the same time point? Is this result then a final equilibration, or is it a steady state and you still have something that nucleates and dissolve and
Yeah, no, that's a good question. So they are batch experiments, they are not steady states. And we wait for the system to equilibrate before we do the characterization. So if you use time-lapse imaging, you can clearly see there's a stage where the minerals grow and then they stop growing because you've reached, equilibrium with the solution. And that's when we do the mineralogical analysis. So yeah, that's the end state of the system. And the phases that tend to be unstable, like vaterite, we find that they stay for many days stable, even in solution. So we have time to characterize them and so on. They don't immediately transform into something else.
And this equilibration time is the same for all of the samples?
It will depend on the condition, of course. So you will reach equilibrium more or less quickly. Typically, in the data set I've shown, it will be a few days.
Okay.
Okay, we have an additional question from the chat. It's actually the same colleagues who asked for the first question in the chat. What functional group are present in EPS of the bacteria that you are studying? Are they too electronegative?
Yeah, so for the diatom, it's mainly chitin. And now I'm getting tired. I'm trying to remember, the groups in chitin. Yeah, you could look that up. For the EPS in the bacteria, usually they're going to be a mix of polysaccharides, and they tend to be rich in groups that are negatively charged at neutral pH. So you could have many carboxylic groups and hydroxyl groups negatively charged, and they tend to complex calcium.
Okay. Is there any other question from the audience? There's a new one. Please.
Hi, Julie. It was very nice.
Thank you.
Yeah, I was curious about one of your last slides. You skip it because of time. It was because you generate your library, but I was curious. It's my email I sent you. So on the library and the test, because you need to separate your library for your assay. I was curious how you finish by doing this.
Yeah, so there are the different mutants. So they're growing in different pure cultures. So you can have one per well. So they are separated. And then you make them mineralize in the same medium. And you try to see if the mutations are any effects on the mineral. So you can see it visually on the images, but you can also extract the morphological parameters and do things like PCA, for example, and see the mutants if they cluster in different location from the wild type. So this is the slide as I had to skip. But what I wanted to say is that it's not so straightforward because these different mutants grow at different rates, and the cell density has a huge influence on the mineral properties. So we need to disentangle those different effects to really better understand what's going on with the mutants.
Yeah. I was curious technically because you start with your library, like you do the metagenesis, and after you go for the pure culture. How do you come from the pure culture at first? Like you select.
Yeah, you plate them. You plate your mutants and you isolate single colonies. That's how you separate them. Yeah.
And how much did you select? I'm just curious.
So Mohammed so far has 71 mutants.
Wow.
Yes.
Cool.
Yeah.
Thank you.
Okay. Any other question? I don't see any question. Oh, maybe this one just arrived. I was wondering if you know whether the negatively charged EPS groups preferentially bind divalent cations like calcium versus
Magnesium.
magnesium, I guess, yeah, or others.
Or other. Well, yeah, I think this is known. Like you can look at partition coefficients for different cations on different types of organic groups, and so I don't have this type of results in mind, but I'm pretty sure there are studies that look at that specifically. This is not something I've done.
This is somehow part of the question I had in my mind. I mean, we can still have the crystal structure of calcite, so without any, let's say, significant change in the structural models, even if a fraction of magnesium, we are talking about magnesium, is into the structure. And obviously, you can have different level of ordering or disordering, if you prefer, between calcium and magnesium into the structure. I think that it will be an additional, additional parameters to be considered, but it can play an important role in the final morphology of the crystals. I mean, at least the experience of people involved in crystallography is going in this direction. So let's say the fraction of minor elements, subordinate elements, at the level that they are not able to change the structure significantly, but can have an influence on the morphology of the crystals. That's something that should be considered.
Yes, I agree with you. And in the environment, it's going to be an important parameter. It's also going to control the polymorphism and so on. So
yeah, clear.
In
Clear.
this larger project, we will do one as a follow-up. This is one of the parameters we want to test, concentration of magnesium. Absolutely.
I would not be surprised that even substitution at, let's say, hundreds ppm level can play a role in the change of the morphology of final crystals. I will not be surprised for that. Yeah.
Yeah, that will be interesting to see. Yeah. Thank you.
Okay. Isabel, I don't see other question.
Yeah, maybe we can all have propose that we have a round of applause to Julie, and congratulations for this award and the medal.
Well,
thank you for driving us into this wonderful world of the calcite minerals. How a single composition at bulk can be influenced by all these parameters and be so different depending on the condition. Thank you very much.
Well, thank you very much, and also for the opportunity for the talk. Thank you.
Okay, so we can say that this public lecture is concluded. And
thank you, thank you everyone for attending. That was really nice to see so many people being involved.
And obviously, you will we will get informed if any other activities, opportunities promoted by the European Mineralogical Union in terms of lecture will be promoted later on by your own national societies. Okay. So thanks a lot.
Bye-bye.
And bye-bye.
Bye-bye.